Hello, everybody, and welcome to Ignite. This is the first set of Ignite talks we've held here at MSU. I am Dr. Abby Stevens. I am the Public Program Manager at the MSU Museum. And it has been my privilege to work with 9 research rock stars over the last 6 weeks or so and help them get ready to give Ignite talks this evening. I'm going to give you a quick primer on what is up ahead. So Ignite is this thing where you have 5 minutes, only 5 minutes, to get across one big idea. So this means that you get 20 slides and they auto-advance every 15 seconds. Has anybody ever given a presentation with slides that advance beyond your control before? Ignite people have, but that's a different thing. So Ignite started in San Francisco in 2006, so it's been around for a while. And since then, it has been in 350 cities across 6 continents. We are the first one here in Michigan, which is very exciting. And we hope that this is the first of many. So at MSU, we had an open call for students, graduate or undergraduate, doing a research project in any field. So we have a fantastic lineup for you. Previous public speaking or stage experience was not required. And we selected 9 research rock stars to talk to you this evening. And we had a research communication workshop to help get everybody ready. So if you sign up for this in the future, I do not throw you up on stage without any preparation. I promise. I'm very nice. And we are looking for more students in the spring. You'll have more information about that at the end, but Ignite in the spring is going to be March 14th. So we would love to see all of you back then. I am now going to turn it over to Natasha T. Miller to MC for the evening.
Give it up to Abby for putting this together. Is this mic work? Okay. So I'm wearing a mic for no reason at all. But yeah, give it up for the researchers, the 9 researchers that you'll hear tonight. I am Natasha T. Miller. I'm the Community Engagement Manager at the MSU Museum. I am not a doctor, so I might not understand anything that's happening tonight, but it is my job to make sure that you all are here and you all are present. We like to say in poetry slams, if you enjoy anything that these students are saying while they're on stage, you can make some sort of noise. No noise that is like distracting, but if you've ever been to a poetry slam, you want to snap a little bit, you want to do a little bit, just to let them know that you are enjoying what it is that they're saying. They put a whole lot of hard work into these presentations, and I'm really excited to see each and every one of them. And make noise for the first one, as you will for the last one. Don't just clap because it's like your kid or your sister. They don't win anything but the love of the audience. So yeah, you will hear 5 amazing researchers. Then you'll hear this amazing jazz band again. Give it up for the jazz band. You'll hear 4 more researchers, and then you'll hear the jazz band again, and then you can talk and you can mingle and you can get brochures for the events that we have happening over at the MSU Museum. You can talk to myself, you can talk to Dr. Stevens, and just figure out where we go next. So are you all ready for your first researcher? Okay. No, like, are you really ready for your first researcher? Like, make a lot more noise than that. All right, everybody welcome Brett to the stage. See how I'm not the mic. There's 2 of them. Yep, there's 2 of them, see? This is not properly researched.
Do I just take one?
Nope, you take both.
Oh, it's both of them.
Okay. One is for the room and one is for the recording.
All right.
Give me the key when you're ready.
Before I was even born, my grandfather died from a heart attack. This is when my father was only 7 years old. My name is Brett Volmert. I'm a PhD candidate in biomedical engineering, and today I'll be telling you about growing mini hearts from stem cells. All throughout the world, heart disease is the leading cause of death, with over 18 million people dying each year from heart disease. That's over 50,000 deaths every day. Furthermore, congenital heart defects are the most common type of birth defect in newborn babies in the United States, with over 40,000 cases of newborns having heart defects each year. There are so many unanswered questions surrounding heart disease. Traditionally, scientists use animals and cells to investigate heart disease and to try to understand why they occur and how to treat or even prevent them. Unfortunately, these models are not fully faithful in fully capturing the disease found in human hearts. Now, animals and cells are problematic for a number of reasons. Animal hearts have different structure and function compared to humans, and in cells, there are just too many missing pieces to the puzzle. Scientists and doctors require better tools to properly and fully investigate human heart disease. What if we could grow hearts from scratch? What if we could mimic the events that happen in the embryo in the formation of the heart? Well, that's what we're doing. We are growing mini human hearts from stem cells through mimicking the events that happen in the embryo. These mini hearts have a complex network of blood vessels, they beat, and they have all the main cell types that are found in the heart. When I say mini, I really mean it. These hearts are less than half the size of a grain of rice. Even so, they are still incredibly powerful tools that can help illuminate the factors that contribute to heart disease. Now, all of this surrounds around the power of stem cells. Stem cells have the ability to turn into any type of cell or organ in the body, and we manipulate this power to grow mini hearts. So how does this work? Stem cells have a type of genetic map inside of them, and we simply tell them which way to turn. We start with a clump of stem cells, and we mimic the events of the early formation of the heart in a dish. And this can provide superior and more detailed insights surrounding heart development in humans that has ever been studied before. These mini hearts have electrical activity and can beat, allowing us to investigate electrical and rhythm disorders such as arrhythmias in humans. Furthermore, these mini hearts have a complex network of blood vessels, which serve as a platform to investigate the role of blood vessels in various types of human heart diseases and circulation disorders such as atherosclerosis. Similar to how the human heart has atria and ventricles to pump blood throughout the body, our mini hearts have similar structures. This level of complexity can help us to illuminate the origin of many types of human heart diseases. In this way, we've shown how a common drug that pregnant women take during pregnancy, known as Zofran, can contribute to heart defects in newborn babies. These mini hearts can be imaged using MRI and CT to show us how these hearts are growing over time and how large or small the chambers are inside of the mini hearts. And what makes all of this even more incredible is the technology known as induced stem cells. We can take a sample of skin or blood from any human and turn that into stem cells. And we can then turn that into mini hearts. And we can do this for any of you. We can take your skin or blood, we can turn that into your stem cells, and we can make your mini hearts. We can create thousands and thousands of these mini hearts to investigate your disease and to help find more effective treatments for your heart disease. We are embarking on a new frontier of science and medicine, and I am so grateful to have been able to share it with you all today.
Thank you.
Give it up for Brett again. That talk made me feel like I'm have like a new heart by the end of this session. Like take my skin cells on the way out the door. It's like, oh, okay, thank you, appreciate that. I needed that. All right, are you ready for your next speaker? All right, make noise for Shahad.
Hello, everybody. Not everything in life is black or white. Sometimes the best parts of life are gray, or whatever colors you choose to blend to design a colorful and inspiring character. My name is Shahad Nasir, and today I'll be presenting my research focusing on culture's impact on identity and affiliation. To start off, I would like to ask how many of you have lived in a country with a different culture, or at least believe that they belong to more than one culture? Okay, it's expected. Many immigrants come to the U.S. hoping for better education and work opportunities, but the younger generation struggles to adapt to the environment or the American culture. The purpose of my research was to highlight the key struggles of belonging to more than one culture and address ways to deal with these problems. I focus on third-generation MSU students from Muslim immigrant families and analyze ways of how they adapt, resist, or merge the different cultures they belong to. A point to highlight is that the findings from my research does not belong to only a group of people. It's like it belongs to people from different backgrounds, setting aside religion and other classifications. With today's ongoing globalization and multiculturalism has become very common for a person to belong to at least two cultures, especially here in the US where there are people from diverse backgrounds. But there's always this question of that you don't belong fully to either culture, the question of where do I belong. This slide presents the names of my participants, how many years they have been living in the US, and what cultures they belong to. I'll be stating the results of my research, and then I'll be providing an example of how it applies to me as a student from an immigrant family from Iraq living here in the US. This is a book that I have read recently by Gordon Matthew. It stated that it's a cultural supermarket where there are multiple cultures and it's impossible to identify to only one culture. You'll always find parts of your identity that blends in with different cultures. Many of my participants indicated that they were caught between both their American culture and their culture of origin. The longer they lived in America, the more they adapted to the culture. And then they learned to embrace things that makes them different while also preserving things that they had in common. I come from a multicultural background where my family is originally from Iraq, but I've also lived most of my life outside my country, Iraq, in Syria and Jordan. But all of these countries shared a similar culture. So when I came here in the US, I had an identity crisis where I found the culture very hard to adapt to. I tried to preserve the culture as I grew up into, but also was open to learning about new cultures and basically absorbing parts that I believe fits well into my identity. You can try, for example, I learned about the American culture through classes I've taken before high school or currently in college. I have friends from different states, and I tried to blend in by celebrating some American holidays that are usually not part of my culture. For example, Thanksgiving. Many of the participants indicated that they tried to stay connected to their culture by presenting their non-American identity and trying to be involved in events that best presents their culture. They attend cultural events, eat food from their country, wear traditional clothes, and then just trying to identify a place for them. And then many of the students indicated that MSU has more than 4,000 international students, which emphasizes how important this struggle is. It's incredibly hard to leave behind your familiar way of life and come to a place where it's hard to find where you belong. I preserve my culture by being a member of the Iraqi-American Union, the Arab Cultural Society, and by forming a social circle where I share my culture with other people. Sometimes it may be hard to connect with your roots because you might not have had the same experiences growing up as the same as your culture, or you might not have a good exposure because you did not spend as much time in that country or the culture. But it's always important to preserve that culture because this is what's being passed on from one generation to the other. One of the key struggles that my participant identified was not being able to use their first languages often, thus usually gaining an American accent, which makes them identify as a minority in their culture and more as an American. So learn about new cultures, but always preserve the cultures you were born into. It's okay if you do not belong to any culture. Create your own culture by combining parts of other cultures that fits well into your identity. Never try to conceal who you are and adapt to what you want to be and what you love. Adapt yourself to what you want to be and what you love. I don't belong to only one culture. I'm not only this or that. I belong to many cultures, but because my family has always exposed me to the Iraqi Muslim culture, I feel like I belong more as an Iraqi than other cultures. And here's where the art comes to play, where you can where you can blend in your identity to something where you are most happy with and best represents your values. Be proud of who you are and don't stick to only one culture because it will for sure set you with limits. Use your multicultural background as a gift and blend in colors to design your identity. Combine cultures to thrive. Thank you.
Keep the love going, Shahad.
Thank you.
Before we get into our next presenter, I want to make sure that we show some love to ARCA. We're in their space, so give it up for ARCA for allowing us to be here as partners. And now we are on to our next presenter. I'm used to saying performer. I'm just going to perform at some point because y'all are so good. It's like my turn now. But yeah, give it up for Hannah. Wow,
You got it.
Thank you. Need 2 pockets. Oh dear.
Okay. Give me a thumbs up when you're ready.
Thumbs up.
Okay.
Hello everyone. Today I want to tell you about how I create tools to help breed fast cooking dry beans. So my name is Hannah Jeffery and I'm from the College of Agricultural and Natural Resources here at MSU. And you may know beans in the form of pinto beans, black beans, whatever you get at Chipotle. And beans are an opportunity and a challenge because they are an affordable source of plant-based protein. They're healthy for you. They have high levels of zinc, iron, calcium, folate, dietary fiber, so many good things. However, they take 60 to 90 minutes to cook on average, and the cooking times that they have are widely variable. So, how do we tackle this problem? Make beans more accessible to the average consumer. What if we could make beans take only 20 minutes to cook? Make them have shorter cooking times, take less of your time, help the environment by using less energy. What if they could cook in about the same amount of time that it takes you to cook your rice? Rice complements beans in their protein content, and so it makes the perfect protein. Simple meal, fast, easy, out the door. So, what we propose to do is help to lower their cooking times using something called breeding. And in order to do that, we have to know what their cooking times are. We use something called a Matson cooker, and while it is reproducible, it's an incredibly expensive setup. It's not easy for everyone to do. What I propose that we do is that we improve the totality of the plant and helping to know which plants to choose, which plants to breed together, helps speed up that process. You can think of what I'm doing as basically just looking at the template of life and picking out only the parts that are relevant to cooking time in dry beans. So for example, I want this particular building block, this allele, one could call it, because it decreases cooking time or has no effect on it. I'm going to put that into my creation. You could also, if you're a painter, you could think of it as mixing paints together in order to create just that perfect blend that you want to see in your final product. And I'm using that as a tool. You don't have to worry about all the funny names there. That's just some bioinformatics stuff I use. But basically what all that is doing is it's acting as a funnel, as a filter to take many, many genes, millions actually, to thousands, to hundreds, to hopefully just 3 or 4 that are important and that we can use in order to successfully lower cooking time in dry beans using breeding. So, what are our puzzle pieces? Well, we're still working on that, but we do have some candidate genes, which is pretty cool, that seem to have a significant effect on cooking time. And we've also discovered a lot about the processes that control cooking time in dry bean. You don't have to worry too much about that. I'm going to summarize it for you. Watch. The cooking time factors that control cooking time include cell walls. The thicker the cell walls are, the longer that they tend to take to cook. And what's next is their permeability. So if the seed coat, you know, that pretty bit on the outside that protects all of the good nutritious stuff on the inside of the bean. If it can't let in water, then the seed won't cook properly. And finally, calcium content. If the calcium content of your beans is high, or if the calcium content of your soaking water or cooking water is high, then your beans are going to be rock hard. So while we're working on breeding beans for better cooking times, what can you do as the average consumer to save yourself some time? You should, 1, soak your beans for 10 to 12 hours. Preferably overnight because that's easiest. Use a pressure cooker. It's actually much better for the environment to use a pressure cooker to cook large batches of beans at once. Then you can freeze them, have them for the rest of the week. And finally, you should eat them as fresh as you possibly can. Try to store them in a dark, cool, and dry environment if possible, like your pantry. So thank you very much for listening to my spiel about beans.
Excellent job, Hannah.
Thank you.
Thank you. Make more noise for Hannah. I'm not sure what your Thanksgiving plans are, but you can get it spilled to my grandma the morning of, so we can eat before 10 p.m. That'd be great. Yep. Make noise for your next presenter, Ceco. And if you're enjoying the talk so far, make a lot of noise right now. Like, let them know.
Yes. Okay. Hello, everybody. My name is Ceco Maples. I'm an undergrad from the Department of Anthropology, and today my presentation is on qualitative evidence synthesis for Indigenous health research. So first, I want to define what evidence is. So I'm going to talk about 2 types of evidences, but evidence comes from studies that experiments and observations that use the scientific method. So the first type of evidence is quantitative research, or research that uses numbers. It can be applied, quantitative research can be applied to large populations, and it's a bit technical sometimes. And then there is qualitative, or research that incorporates emotions, thoughts, and feelings. There's a lot of misconceptions about it. A lot of it is talking to people, but it can be more technical than that. So both types of these evidences have their own strengths and weaknesses. So a strength with qualitative or research that includes thoughts is that it can include knowledge from different cultures, and with quantitative or research with numbers, it usually has more weight in public health decisions, but they also have their cons too, as qualitative or research that uses thoughts, emotions, is less prestigious. But although I'm talking about it as it's a dichotomy, they're actually 2 sides of the same coin, which is to inform clinical practice, in health research, inform clinical practice, policy, and more research. An example is qualitative research, informing how your therapist talks to you, or research that uses numbers determining COVID mandates. So I want to know how these evidence applies to the Indigenous peoples of the Americas, which is about 55 million people, but it can also be applied to potentially billions of people who are affected by colonization. So the effects of colonization is sometimes overlooked when it comes to the health, how it affects health. It affects health in multiple ways, and research and colonization have a long intertwined history. So evidence has long been used to justify scientific racism and genocide, but even though we've come far in the 21st century, there's still a lot of problems when it comes to research on Indigenous peoples, such as not consulting with Indigenous peoples or not having Indigenous peoples on any part of your research. And this can lead to a lot of problems because evidence informs clinical practice and policy and even farther research. So I introduced evidence synthesis as a possible potential solution to colonized research, as it looks at, combines multiple studies, themes from multiple studies that have the same outcome, and it can be used to apply to bigger problems such as colonialization, and there's multiple ways that it can be applied. For example, it can be applied to mental health, maternal health, and nutrition, for example, because those require a lot of traditional knowledge. But overall, it's not a competition between which type of evidence is better, because they both have the same goal of health equity. So I would say that there needs to be more collaboration between Indigenous communities, research teams, and policymakers, because racism and how it affects health is a bigger issue. That's just not researchers' responsibility. So I would say that qualitative evidence synthesis is not the solution. It's not a replacement to research with numbers, but it can be used to inform and complement research that uses numbers, quantitative research. But it shouldn't stop there, and we should also decolonize more aspects that prevent Indigenous peoples from accessing healthcare and what causes health inequality.
Awesome job, Ceco.
So we only have one more presenter before the band comes back on. It's not necessarily a break, but if any of y'all have, you need to use the restroom, you just go right out those doors, make a right. Feel free to do that before the next half of the show. I love that talk. I love anything that informs how your therapist talks to you, because my therapist just stopped talking to me. She's like, I have no words. I'm like, really? Why? So I might take you along too. All of y'all just come with me when I go back home. Again, we are the MSU Museum. You can come visit us anytime during business hours. It is completely free. We definitely have a lot of great events. We talked about our First Fridays that we do. It is a safe space. It is a fun space for students to come and hang out. Our next First Friday, we're actually having a silent disco. Last year we had one. It usually gets around 250 to 300 students. We bring in some amazing DJs. This time we have John Collins from Underground Resistance in Detroit, who is one of the founders of techno music. So students come, they put on these headphones, you switch between 3 channels. We provide free food, again, a safe space. So come check us out the first Friday in December. Are you ready for your next presenter? Make more noise for Andrew.
Can y'all hear me? Yes. Yeah, okay, we're good? Yep. All right, okay. And we're ready. Hi, my name is Andrew Hu. I use they, them pronouns, and I'm here to talk to you today about why I think that K through 12 computer science education should have less code and more focus on community. So if you've never taken a K through 12 computer science class, it's actually a lot more like woodshop than it is a math or a science class, and that's because they're trying to teach you practical skills to help you get a job, to write computer code, to create programs that are the foundation of all the technology that you're interacting with. And these decision-making processes are what we call algorithms. They're basically the building blocks, everything that's behind all the technology that you're interacting with, and that's where the decision-making happens. So the people who create those, the decision-making processes, the algorithms, are influencing the effects of technology. And those algorithms are changing the world both for better and for worse. For example, we're using technology to help find new cures for diseases. But we could also use it to create a surveillance state using facial recognition, and that's happening right now. That's bad stuff, right? But what's often left out of the conversation is this thing called justice, okay? Justice isn't just about doing a good deed, it's about changing the system, okay? And that work is going to take a lot of systemic fixing the system, but right now, algorithms are doing more injustice than justice. Here's an example from here in Michigan. The state tried to create an algorithm to detect unemployment fraud. It was wrong. It hurt tens of thousands of families when they were hurting the most, when they needed help from the state. And that was an injustice created by an algorithm. Right now, there's a huge growth in demand for computer science education, both in college, and I want to focus on K-12. And that's because families believe that K-12 computer science education is going to unlock lucrative careers for their children. But I think that we should have less code and more community, focus on community in our education. 3 reasons: making informed decisions, organizing as a community, and creating more inclusive, interdisciplinary classrooms. Okay, the first is these CS classes are not for your CS majors, okay? Look at how many people are in high school versus how many people are going to become programmers. But all of those students in CS classes are going to be interacting with technology on a day-to-day basis. So they need to be able to make informed decisions. Here's an example. Life 360, an app to help track your loved ones. Turns out they were selling our location data. If we know about these practices, we're going to change our behaviors, right? We need to help students make informed decisions. So how that could work in K-12 classes is that thanks to EU data privacy laws, we can now look at what data is being collected on us. Here's one from Facebook. We can have students look at what their data is and how that might influence their behaviors, help them think about that. Next reason, schools are the foundation of democracy. We're preparing students for voting, for organizing, for using their freedom of speech, and they should be doing that with computer science content too, right? So right now, there's a big challenge with ChatGPT. It's turning the whole world upside down, right? Students that's a big problem in schools, right? Students should be part of that conversation, especially in computer science classes. They can have a debate about the pros and cons and even take those results to the school board, make a real impact in their community, right? But the current system doesn't see that as a value. The way that I see it, looking for errors in code while ignoring the real-world impact is like looking for spelling errors in the Constitution while ignoring its real-world impact on us, on our day-to-day lives, right? Okay, so of course, CS has a huge diversity problem, right? But part of the reason that I think that's happening is because the only way of thinking that's being valued in computer science is programming. But if we expand that, if we be more inclusive to many different ways of thinking, I'm talking about collaboration and debate and communication, then students who don't necessarily fit the stereotype are going to feel more welcomed and they're more likely to see themselves in a computing career. Okay? Another way that we make it truly inclusive is that we center the work of women and scholars of color who have been talking about this even when it wasn't popular. Like Timnit Gebru, who got fired from Google for speaking out, and Emily Bender, who's been studying ChatGPT for tools like ChatGPT for years. All of this together is what we call justice-centered computing education. We're connecting with students' communities. We're having them be critical in the sense of critical race theory to understand how algorithms are upholding systems of oppression. And we're also preparing students to be citizens. So my work is connecting K-12 CS teachers with that justice-centered computing and the real-world examples so that we can co-design lesson plans that are going to work for them in their classrooms, making it relatable to their students. Okay? So to sum it all up, the reason why we need less code and more community in our computer science classes is to make them have more well-informed users of technology, have a stronger democracy, and have more inclusive classrooms. Thank you.
Make more noise for Andrew. Hope you all had a lovely break connecting with each other. Again, we encourage you to speak to these researchers after the show, find out more about their research and their inspiration. Are you guys ready for the second half of the show? Also, we have a lot of amazing people watching this on live stream, so make a lot of noise to show them how much fun you all are having in this talk in person. So. All right, so give it up for our first performer of the second half. Make noise for Cathy.
Hi, my name is Cathy Mercado. I'm a second-year PhD student in the Department of Plant Biology, and this is my talk, Bouncing Back from the Brink, The Secrets of Resurrection Plants. As a plant biologist, I wouldn't say I'm very good at taking care of plants. I often neglect them quite a bit by forgetting to water them. Here's an image of one of my plants currently. But I'm not the only one who's pretty neglectful when it comes to watering plants. Drought is one of the biggest concerns when it comes to our crops. With climate change only exacerbating the current effects of drought stress on plants, this will continue to inhibit and increase impact on our global food supply. With that in mind, we need to think about crops and plants that are more drought tolerant. With plants typically dying at about, after losing 40% of their relative water content, we need to rethink of how we think about plants. What if I told you there was a plant that could lose 90% of their relative water content and still come back to life? Fittingly named resurrection plants, these guys can go dormant for about a few months to even years without water. Scattered throughout the plant kingdom, these plants have enormous amount of mechanisms to deal with water depletion and extreme drought stress, also known as desiccation tolerance. Looking at this video that's about to play, hopefully, this is not playing in reverse. These plants can restart their metabolism or their day-to-day processes rather quickly in from a few hours to just a few days. The slide is messed up now.
That's okay. It's good.
Okay. The adaptations of these plants are enormous. And what's critical about this is understanding how they've survived with the amazing amount of plant resilience and how we can adapt these strategies to future plants. But the question is, how and what traits have these plants used in order to adapt to a life without water? Plants and life evolved around water, so my job as a scientist is to study these anomalies. I'm focused in on the tiny cellular and macromolecular instruments that these plants are using in order to survive desiccations. And I'm about to take you on that journey with me. As the plants are undergoing their drying state, they begin to turn their machinery on into desiccation mode, creating proteins necessary to survive this stress. And in that time, once they're dried, they begin to hoard these mRNAs that they're turning into proteins for later regulatory use when they're rehydrating. And this is important for restarting the plant metabolism up again. But the plants are also reshaping their cells in their cells. The process of them losing the water in their cells helps them better be equipped for this low moisture environment if they're in a state that's a lot smaller. At the same time, they're also destroying themselves in ways where they're keeping their photosynthetic machinery at bay to avoid oxidative damage. This can be in the form of destroying their chloroplasts or increasing their antioxidants. So kind of like, and then they enter this frozen state, kind of like Avatar Aang in the hit series. They're waiting for a time when it's time to come back. But in order to do this, they use sugar instead of water to create a frozen state. This frozen state isn't exactly like ice as it's a uniform crystal. It's more of like a glassy solid. And as the water begins to deplete, these crystal structures are able to reform. Now that we kind of know some of the actors, we're taking all these plant elements to help better understand desiccation. We know that what they are, but now it's time to figure out how they're being used. And this is important for re-engineering plant species. If we have more desiccation-sensitive plants, they're not able to do the same things as our desiccation-tolerant plants. And without over-bombarding them, if we re-engineer them with traits, we have to kind of be in that fine-tuned balance without harming them so that they can be more drought-tolerant. For instance, there's a cousin kind of the resurrection plants. This is a Ethiopian grass teff, which is very nutritious, and we can help look at traits that are very similar to our resurrection counterparts. And by doing this, we're able to really think about how we can apply genes and traits from related resurrection plants to crops for a very better and more sustainable future.
Nice job.
Give it up again. You really can't take care of plants? Wow. I have a money tree at home. And when I used to water the tree, I had money. Now I don't water it, and I feel like if you look at me in the corner and I'm drinking water, like I'm going to keep it up. So, help. Are you ready for your next presenter? Make noise if you are.
Hi, my name is Alva, and I'm a sophomore majoring in information science and in the Department of Media and Information of the College of Communication, Arts, and Science. And today I'm going to give a talk about the thinking of thinking. So technically, I think this green bird is very familiar to most of you. And it's actually, if you're joining the online language learning community, you will be familiar with it. And it's the same with me, because actually, I have learned Duolingo I have used it for 3 years. Since July 2019, I have a lot of diamonds, a lot of achievements, a very long streak. But there's a very initial problem that I cannot speak French even after 3 years I'm using it. And I start to question whether it's my fault or it's the bird fault. But yeah, I feel like if I keep asking this question, it doesn't help me a lot, so I reframe the questions into another thing. The question is that, so machine has learned so much from us, yet what can we learn from it? By thinking about the way we think. So that's the process. I come up to the concept of metacognition, which includes meta, above, about, and cognition, to know, to think. So for a basic example, when you listen to Ignite talks today and you think about specific topic, it's your cognitive process. But when you link it to other topics and you reflect how deep you understand a topic, it's metacognitive process. So if you link my topic to Andrew's topics about computational, you're in the metacognitive process. And this concept has been studied by so much scientists that I cannot read the terminologies, as you can see, but it also shows a part of the importance of the concept in today's eras. When you interact so much with technology, when you learn and do so many things with technology. And this concept includes two components. It is metacognitive awareness and regulations. Basically, awareness is how you're aware about the way you learn, and regulation is how you change the way you learn. In the human context, metacognitive awareness is super strong because we had a tool named 5H questions. You ask how, why, what, when, where you do anything. You have a strong awareness, but you lack of implementation. So for example, I know I did not learn so well about French on Duolingo, but I cannot change the way I'm learning. There's some issue. But in the computer context, the computer actually really good at planning, monitoring, and evaluating stuff, because they have specific algorithms designed by computer scientists. But they lack of transcendence, so they have weakness of experience the world by the way we experience. I'm thinking about the way we can use the strength and weakness of both human and computer by cooperation for innovation, the hybrid thought. So when you come to the way humans learn a language, you have the mechanism of the sphere of meaning. You understand a new word of a new culture by incorporating it into different contexts of the cultures. That's how you learn a new word in the language. But for the machine, they also have a way of learning called the word embedding. So just imagine you have a piece of logo, but if you want to build a whole models, you need all the pieces. And if you don't have all the pieces, you cannot see the whole models. That's the way the computer learns about words. So applying it to Duolingo case, I want to quote a very famous linguistic, John Rupert. You shall know a word by the company it keeps. If you want to learn something, you should combine the methods of learning. So for example, you can take the word from Duolingo and find it in poems, songs, movies, in cultural context. I do have a very cool method. You can take words from Duolingo and put it in ChatGPT, ask it to write a poem for you. It is the best way of learning, I would tell you, because it's so effective for me. So I would say a conclusion that Duolingo is really good, but it's not enough for your language fluency. And if you really want to improve your language proficiency, you might need to think about the way you learn it. So 2 takeaways that I would love you to bring back to home is that you should reflect more on your learning methods in this human-computer interaction era, because we can utilize technology a lot for the way we learn, but we need to reflect deeply about it. You can also learn from others and also from machines. And most importantly, if you want to be a participant of the research project I'm conducting right now about this topic, you can scan the QR code. It includes the sign-up forms and also the references and the reading materials that you want to figure out more about the topics. So it's also time for your daily lesson. If you haven't learned Duolingo yet, I would highly recommend it. Even though it's not really effective for me to learn French, but it does motivate me a lot to keep me going up and going on with my language learning journey. Thank you.
Okay. Start clipping down. Thank you.
Aside from that talk, keep it up for this great shirt.
Nice shirt.
So we only have 2 more performers. Everybody say, "Aw." That's so sad. I feel like I should have paid $365 to be here. Y'all are that good. And you all really know. 65 though. You know what? I have no idea why I said that. I'm just learning from the research. I'm saying things now. Everybody make noise for Mariam.
How can we detect harmful bacteria and prevent illness? Hello everyone, my name is Mariam Shahab. I'm a student here at Michigan State University. I worked in Dr. Debiji Saha's lab this past summer, and I'm going to walk you through the idea that insects can smell bacteria. To begin with, there are 2 main states of bacteria, planktonic bacteria and biofilm bacteria. Now, planktonic bacteria tend to be less dangerous because the bacteria lives alone, but biofilm bacteria is more dangerous because the bacteria live together. And because of this, they're more resistant to antibiotics. In fact, in humans, about 80% of infections are caused by biofilms. And currently, antibiotics are becoming more and more ineffective. Now, the 2 states of bacteria have different odors. As an example, the planktonic bacteria might have a citrus-like smell, while the dangerous biofilm might have a grape-like scent. And the important thing is both these bacteria smell different. The dangerous bacteria can be identified. Now, what animal has a strong sense of smell? They're usually used at airports. It's dogs, right? Dogs have an incredibly powerful nose. They can tell if a person has a disease or not. However, it takes time to train dogs, and it's expensive. Now, insects, just like dogs, have a powerful sense of smell, particularly locusts. Locusts are just like grasshoppers. Grasshoppers tend to be solitary. Locusts live in huge swarms, and they destroy crops. And that's why farmers don't like them. Now, apart from being infamous crop destructors, locusts have powerful antennas. Their antennas are analogous to nose, so they can smell with their antennas. And that's how they find food, mate, and avoid predators. So they have a strong sense of smell. In fact, my lab recently showed that they can detect cancer. They can smell different cancerous odors. So if they can detect different cancer odors, can they detect different bacteria? This is what we investigated this summer. I helped run the experiments, and I also got to learn more deeply how the locusts detect bacteria. So in the brain of a locust, we have a special region called the antennal lobe. This part of the brain processes information about odor. And the brain is made of cells called neurons, and these neurons communicate with electrical signals. The electrical signals is what we're interested in. So we use a tool called an electrode to record those electrical signals. And this is our recording. The blue rectangle basically shows when the odor was delivered, and the red trace you see, those are spikes. Now, what is a spike? A single spike is called an action potential. And coming back to this recording, we can see that there is a change in the pattern of spikes. This means that the locust is detecting bacteria. Now, for our experiments, we used 2 species of bacteria, and for each species, we used a less dangerous state and a more dangerous state. So we gave the locust the odor at the antenna, and we got recordings. These are our recordings, and we can see that the spike pattern is different for each odor. So we used software to process this data, and we got a much cleaner result. This plot shows that not only is the locust detecting the bacteria, it's responding differently for each bacteria. So, and we can tell the difference between dangerous and less dangerous bacteria. Now this is just one data set. We repeated the experiment multiple times. We collected the data and made a template. You can think of this template as 5 unique fingerprints. For every odor, there is a unique fingerprint. When we present an unknown odor, so an unknown fingerprint, the template will try to find the closest match. This is the working theory behind how we visualize what's called a confusion matrix. This is our confusion matrix. On the bottom, we have our unknown odors, and on the side, we have our odor template. The dark diagonal shows that most of our odors were correctly classified. We achieved an accuracy of 76%. To conclude, locusts are sensitive to odors, and our preliminary data shows that the locusts can detect different bacteria. They can distinguish between less dangerous and more dangerous bacteria. For future work, we hope to collect more data and improve our template. And we also hope that one day this cyborg locust will be used to test human breath for dangerous bacterial infections. I'd like to thank Dr. Hardy for helping us make the bacterial cell cultures. Thank you.
Can I do a microphone? Oh, yeah. Thank you.
I love how you said confusing, and then Abby made it confusing.
I know.
Okay.
We are on to our last presenter of the evening. We really appreciate you all being here. You are the rock stars as well. This was a rigorous process, so I want you all to take for granted the fact that your applications were the most exciting and the most intriguing to the whole team. So give it up for you all again. Now for our last presenter, give it up for Dylan, everybody.
Hey y'all, my name is Dylan Warner. I'm a master's student in the College of Agriculture and Natural Resources, and today I'm going to be taking y'all down the yellow brick road of sustainable agriculture. This is truffles, trees, and tree nuts. Oh my. So, truffles evolved to live underground. So instead of dispersing their spores with rain or wind like a mushroom, they rely on their beautiful aroma to attract animals and dig them up. So, if you find truffles out in the woods, you want to look for animal diggings and the ectomycorrhizal hosts. These hosts are trees that the truffles are in a mutualistic relationship with. Also helps to have a dog or a locust. So, what is a truffle? I mean, did I just find a truffle? Is it filled with ganache? Yes, you found a truffle. Good job. No? When you cut it open, does it look like that? Yeah. You found a truffle. Good job. All right, so going back to that mutualistic symbiosis, think of it as like a handshake underground between the truffle and the tree roots. They're trading resources like water, nitrogen, carbohydrates, and animals also dig them up, and the cycle continues. But my research is on the American chestnut. This chestnut was once coined the redwood of the East because it could reach heights of 100 feet tall, and its wood was extremely useful. It could be used on a lot of things. Its nuts were also very culinarily versatile. They'd be roasted, boiled, ground into a flour for baking, and they were also a sweet treat. So, the chestnut also had a massive range in the U.S. Made up more than 50% of eastern hardwood forests, and in 1909, the U.S. timber industry placed the value of the chestnut at more than $20 million, until an evil pathogen by the name of Cryphonectria parasitica came. It's a fungus, woo! But it's a necrotrophic parasite that also degrades plant tissue with oxalic acid and slurps out the life soil of the plant. Also has cry in the name. In just 50 years, this pathogen, it first got in the Bronx Zoo, and within 50 years made it to Mississippi, killing all of these trees, reducing them down to stumps. So yeah, chestnuts roasting alive on an open fire. So this displaced a lot of wildlife and sustenance farmers, and communities that relied on the chestnut, they were also displaced. Until, thanks to some brilliant researchers and backcrossing, we have resistant cultivars of this tree. However, there's a bottleneck when it comes to growing a seed and having it grow into a tree. It takes a while. My research overcomes this hurdle by propagating these varieties of American chestnut, growing them up for experiments, and then testing which pairing method increases colonization of tubers on these roots. So this is the micropropagation process. You take cuttings of the valued cultivar or variety you want, you place it in a media and you grow them up. And you can do this with like one plant and get a lot. And this is where we're testing the spores and the mycelial inoculation. On the right, that's the spores and the truffles. We'll blend that up. And the mycelium on the left, it's basically just like the vegetative growth of the fungi. And they both grow on roots, and we're trying to figure out which one does it best. So this is the process of mycelial inoculation. We crack open a truffle, we take some of the good stuff out, and we put it in plates. And it'll grow its vegetative mycelium out, and then we can use that to put in soil and then put our propagated trees into. So they'll have truffles on them. This is the spore inoculation part. What we do, we find truffles out in the woods, we clean them, and we blend them up. And then with that, we pour into soil and then put our trees in. So both like putting fungi in the soil and finding which one does best. So they're a dual crop. You can have truffles, you can have tree nuts, and you have timber. They're low input. You put them in the ground and you wait. And then they're also drought resistant, and they're everywhere in the U.S. And there's a way you can help too. You can take a hike, look for truffles, send them to our lab, the Bonita Lab, and consider donating to the American Chestnut Society. And also, when you dig, watch out for plant roots and cover up your diggings, because then someone will find your truffles. But I would like to give a big thank you to my lab and everyone that made this possible. So.